I'm Paramita Basu. I'm a research assistant professor at the Department of Anesthesiology and Perioperative Medicine, University of Pittsburgh. Today, I welcome you all to the first webinar of Pain Research Forum's webinar series. As you can see, we have a few events that's coming up right now. So the one event would be just a week from today. It's going to be on January 21st. Dr. Stephanie Brooke from University of Toronto, she's going to deliver her first journal club, the Pain Research Forum Journal Club series. The next event is going to be the next month, the second webinar. That's going to be on the Power Over Pain Empowerment Jubilee, a novel community engaged event to conquer pain. So for all of these events, you can see we have the slides going on, so you can use the QR code or the link to register. So one important thing to mention that for all our PRF events, you have to register in order to get that Zoom link to attend to that particular event, whether it's a webinar or it's a journal club. The PRF is active on different social media. So we have Twitter or X, Blue Sky, and currently we are also active on LinkedIn. So please feel free and connect with us with any or all of the platforms you like to. Now, without further ado, I will get started with today's webinar on pain and peptide with three outstanding speakers: Dr. Bradley K. Taylor from University of Pittsburgh, Dr. Rulong Ji from Duke University, and Dr. Donald McDonald Ian from National Institutes of Health. So regarding the Q&A, as you can see, we will have two different modes. So first you can type in the Q&A box and I will deliver those questions to the speaker, or you can raise your hands and then ask the questions directly. So I plan on taking one or two questions at the end of each the speaker finishes the talk. Then we'll come back at the end of all three speakers and we'll take the rest of the questions. Okay, so without any further delay, I will introduce our first speaker. I'm going to stop share the screen. Okay, Dr. Bradley K. Taylor. So Dr. Taylor is a professor at the Department of Anesthesiology and Perioperative Medicine and the Center for Neuroscience at the University of Pittsburgh and Pittsburgh Center for Pain Research. He completed his PhD at the University of California, San Diego, and his postdoctoral training with Dr. Alan Basbaum at University of California, San Francisco. Dr. Taylor is the recipient of the NIH First Independent Research Support and Transition Award and the NIH Independent Scientist Award KO2 from NIDA. His research area involves studying neuropeptidergic NPY mechanisms of chronic pain, underlying mechanisms of acute to chronic pain transition, as well as understanding the molecular neurobiology of pain sensitization and opioid dependence. His work has been continuously funded by NIH since 1999 and includes a history of multiple R01s as the principal investigator. He has published over 120 peer-reviewed research and review articles, including senior author articles in Science and Nature. He's also serving on the editorial boards of several high-impact journals and chaired numerous symposia at domestic and international meetings and served on the dozens of NIH study sections. So with that, I invite Dr. Taylor to begin his presentation.
Well, thanks for that great introduction, Paramita, and thanks for organizing this session. And thanks, Becky, for putting this PRF online. That's really, really fantastic. And Adam for all the support you're giving as well. So I'm going to be talking about a project that I really haven't said much about before. So it's be kind of a first time for me to share a lot of this with you. This is data that was collected largely by my collaborator, Nick Betley, at the University of Pennsylvania. And much of the data from his laboratory was collected by an amazing um, graduate student Nitzan Goldstein, who's now at MIT with Van Wang. So, what I'm going to be talking about is asking these questions about competing needs, with pain being one of those competing needs. And we're interested. Nick has been on this project for 7 or 8 years. I joined him like 1 or 2 years into it. And what's the neurological basis for the prioritization of competing needs? So, how does a chronic pain patient choose between behaviors that will meet survival threats at the expense of coping with pain? So, pain patients will be, trying to cope with pain with, positive behaviors like meditation, or negative behaviors like oversleeping or perseverating about it. How do they stop coping with this and attend to behaviors that really are related to survival, such as eating when you're hungry, or drinking when you're thirsty, or escaping um, serious threats? And so Nick and I were really wondering, what's this? Is there a competing needs signal that suppresses chronic pain? Is this need signal for just one type of survival threat, or is it universal? And so the concept here is in the bottom right of the slide, where we're wondering where Nick really launched this with looking at hunger, but then we also integrated thirst and fear. And so the idea is that these threats will activate an area in the brain, which then leads to inhibition of ascending pain transmission.
Brad, we just want to interrupt you quickly. The audience is saying your screen is really small and you're kind of on the presenter mode. Uh, we just need to
Oh, I
fix.
thought we had that right.
Yeah. Uh,
Okay. So, Adam, you want to lead me through that? Because I thought we were all set.
I don't actually think you're in presenter mode. I think it might be something with our settings. Why don't you go ahead
okay,
and continue and we'll work on the back end and see if we can get it
Okay.
there.
Okay.
got it. Okay. Thank
I
you,
am
Adam.
using a large, I'm using two screens, and one of the screens is pretty large.
Okay, got it.
Okay.
Okay.
All right. So we're wondering what this competing signal is, and how is it interacting with ascending pain transmission. Okay, so um, Nick started this off with Amber Aldahoff, and I was brought into this project when I saw Nick's presentation of his paper that was published in Cell in 2018. And here he introduces a mouse model of competing needs. And he found that, and he focused on the formalin test, and then did a little bit of work with the CFA model of inflammatory pain as well. And so what he did is he food deprived the animals for a day, and then compared these animals with ad libitum fed animals, and saw a really profound effect. Whether he was looking at the formalin test or in the CFA model, he found that animals that had experienced food deprivation exhibited reduced behavioral signs of what I call acute inflammatory pain. Okay, so this is a schematic from his paper, where when animals are hungry, pain goes down. Acute reflexive pain, like phase one in the formalin test, or responsive to a hot plate, are still intact. Okay, so hunger inhibits pain. So what about chronic pain? And this is work that was done. And so what I'm going to be focusing on today is transitioning from the sort of the acute inflammatory pain that Nick originally studied, and which he continues to study, and incorporating the spared nerve injury model of neuropathic pain, which we're doing here in Pittsburgh, and Nick is starting to do in Pennsylvania as well. So what we found is that hunger reduces behavioral signs of mechanical hypersensitivity after nerve injury, um, as well as behavioral signs of cold allodynia. And this occurred not only with hunger, but also with thirst. And so in this case, with this assay, the thirst was a 24-hour water deprivation. But these studies have also been reproduced using polyethylene glycol injection, which also stimulates the thirst pathways. Okay, so what's the competing needs signal here? And we began, and Nick began to look at neuropeptide Y as the competing needs signal that reduces chronic pain. And because he'd been studying for many, many years, arcuate nucleus AGRP neurons. These neurons, almost all of them co-express neuropeptide Y, and they're required to drive feeding. And this really piqued my interest because we had published a paper many years ago now where we found that if we used a TET promoter to knock down NPY expression, we could reinstate neuropathic pain. So here you can see this is a variant spared nerve injury model where nerve injury produces a hypersensitivity and it gradually resolves in this particular strain of mouse. When we knocked down NPY with doxycycline, we didn't really see much for very many days. But when we allowed the hypersensitivity to resolve, and then we knocked down NPY with doxycycline, we saw a big reinstatement of hypersensitivity. And so this launched a whole nother project on latent sensitization and opposing neuropeptide receptor inhibition of that. But I just wanted to introduce this to you now for how sort of we got into this project. So this is suggesting that NPY tonically inhibits neuropathic pain. And so we have a hypothesis that NPY is involved, and then Nick had found that the parabrachial nucleus is maybe a target by which NPY release from the arcuate nucleus could inhibit ascending pain transmission. Because the parabrachial nucleus is a major entry point for sensory information. Prolonged activation can drive a chronic pain state, and it's the first site of integration of many affective behavioral states besides pain. So it's really well located here. And Nick found that if he injected neuropeptide Y into the parabrachial nucleus, he could inhibit behaviors in the formalin test. Now, these AGRP neurons express not just NPY, but also AGRP and GABA, and these also could be inhibitory, but when he injected those agonists, there was no effect. Okay, what about neuropathic pain? This is where Heather Allen, when she was in my laboratory, really became heavily involved. And so what she did is she did the spared nerve injury model. She implanted cannula into the parabrachial nucleus, just like was done in Nick's lab that Amber Alderhoff had done. Then she did the spared nerve injury model, waited 14 days, and injected a NPY1 receptor agonist. Here you can see that SNI produces mechanical hypersensitivity and cold hypersensitivity. She found that the Lupro NPY, the Y1 agonist, largely reduced the mechanical hypersensitivity as well as and partially reduced the cold hypersensitivity. Okay, so this led to our working hypothesis, Nick and I's working hypothesis, that a sensory stimulus in the setting of nerve injury leads to ascending pain transmission through the lateral parabrachial nucleus. And hunger, thirst, and fear will stimulate NPY neurons in the arcuate nucleus or other areas involved in thirst and fear, which then lead to the release of NPY, which then inhibits pronociceptive NPY1 receptor expressing neurons in the parabrachial nucleus. So we asked whether survival threats engage these NPY1R signaling to inhibit signs of neuropathic pain. And so here you can see that we're injecting a Y1 receptor antagonist to evaluate the function of endogenous NPY. And you can see that before the Y1 antagonist is injected, hunger produces a decrease in hypersensitivity, mechanical hypersensitivity. I'm sorry, this is cold hypersensitivity. And the Y1 antagonist reverses that cold hypersensitivity. Similarly, hunger can cause a decrease in or an increase in threshold or a decrease in mechanical hypersensitivity in the SNI model, and that's reversed by the Y1 receptor antagonist. And we saw the same thing when we used the polyethylene glycol measure of thirst. So then we asked whether nerve injury engages NPY expressing neurons in the parabrachial nucleus, and this was done by fibrophotometry by Nissan Goldstein and using stimulus evoked FOS expression by Heather Allen. And Nissan found that in the asparaginal nerve injury models, and this was also done by Heather as well, that various stimuli, mechanical or cold stimuli, largely activate these neurons in the setting of nerve injury, but not before the nerve injury. Heather found that with stimulus evoked FOS, she could see an increase in the FOS expression in Y1 neurons after a cold stimulus or mechanical stimulus, a rubbing of the paw was applied. So are these PBN Y1 neurons necessary for nerve injury-induced neuropathic pain? This was done in separate laboratories, right, using slightly different methodologies, and Nitsan and Heather found the same results. And so they both used a chemogenic inhibition approach using HM4-DIGI. So after injecting the virus, Nitsan set up the model for nerve injury, and she found that chemogenic inhibition of these PBN Y1 neurons reverses mechanical hypersensitivity and cold hypersensitivity. Heather did this with additional controls using MTRA controls, and she also did the FOS study together with behavior. So she confirmed that SNI increased FOS expression, and that was reversed with HM4-DI, so it's suggesting the neuronal activity. The HM4-DI is, or the CNO is decreasing the activity of those neurons. And she saw this similar results to what Nitsan found above. So chemogenic inhibition of these neurons reduces neuropathic hypersensitivity. So we asked if these neurons are sufficient for nerve injury-induced neuropathic pain using a chemogenetic activation approach. And here you can see that chemogenetic activation causes hypersensitivity, mechanical hypersensitivity, cold hypersensitivity, and also condition place aversion. And there's other results that Nitsan and our laboratory have collected suggesting that supraspinal sites are engaged here for the affective component of pain. Heather found a really remarkable study where she went beyond and did repeated chemogenic activation of PBNY1 neurons. So she injected C21 instead of CNO here for 7 days in a row. And that produced a behavioral hypersensitivity that lasted for 8 months after the C21 injections had stopped. Similar thing with cold hypersensitivity. So Heather's working hard to try to understand what's going on here. And this first result is in revision, pain. To finish up now, to tell you a little bit more about these neurons. We were thinking that these PBNY1 neurons may be a subpopulation of a small subpopulation of PBN neurons that are important in this process or this target for competing needs. And it turns out that these neurons are distributed actually across many regions and many populations. So Nick had done a lot of spatial transcriptomics that I'm not going to be showing you today. But he did validate those transcriptomic studies with RNA-scope studies. And you can see that first of all, the NPY receptor expression, you can't see it too well on the slide, but it's expressed throughout all levels of the lateral parabrachial nucleus. So you can see it's expressed in about 10, 20% of neurons across the different subregions of the parabrachial nucleus. That's in contrast to, say, the calca neurons or the pancapillin neurons, for example. So there's this ensemble of pain responsive neurons that are spatially and molecularly distributed across many regions and populations. And if you think about it, this may suggest, or it begins to suggest to us that these Y1 neurons are amenable to they're all involved in pain transmission, and they're amenable to they're located in areas that are quite varied and may be receiving and may be responsive to inputs from different brain areas that release NPY, and sort of becomes this this ensemble or this catch-all for competing need signals. So we're very interested in continuing to study that. And some evidence for that. Unfortunately, I don't have you can't really see the summary here, but a lot of the studies that Nitsan and Nick have done are suggesting that in addition to the arcuate nucleus, which is activated by food deprivation, the ventrolateral paraectoductal gray is activated by thirst, and the subparaphysicular nucleus is activated by fear. And to generate fear, Nitsan used fox odor. I haven't really shown much of that data here because we don't have too much data in the spared nerve injury model using fox odor, but we do plan on doing that in future studies soon. So the current evidence is suggesting that non-synaptic bulk transport of NPY to downstream NPY expressing neurons in the PBN are being released into the PBN and inhibiting pain transmission there. So future studies include looking at the outputs of these parabrachial nucleus neurons, and Nitsan and Heather have both done studies and identified these interesting areas that the parabrachial nucleus projects to. The central nucleus is widely studied. And so especially the CGRP neurons that project there, but also the NPY Y1 neurons project there as well. Okay, so I wanted to just acknowledge this is the overall model that we have. So hunger and thirst fear activates NPY neurons, leading to NPY release in the parabrachial nucleus, and then pretty much gates the ascending transmission of pain. Okay, so I wanted to thank people in my laboratory currently, people, recent alumni on the NPY project were Tyler Nelson and Heather Allen, and they were indispensable in the data that was collected here. But even more indispensable was the collaboration with Nick Bentley and Nitsan Goldstein. Also other collaborators around the world, and my very good friend and collaborator, Sean Ferris at the University of Pittsburgh. So thanks, everybody.
Thank you so much, Brad, for a great talk. So due to time constraint, we're going to take questions at the very end. So we'll move to our second speaker, Dr. Rulong Ji. Dr. Rulong Ji is the William Maxiner Professor of Anesthesiology and Director of the Center for Translational Pain Medicine at Duke University School of Medicine. He also holds professorships in neurobiology, cell biology, and immunology. Dr. Ji earned his PhD from the Shanghai Institute of Physiology, Chinese Academy of Sciences, and completed postdoctoral training at Peking University, the Karolinska University, and Johns Hopkins University. Prior to joining Duke, he served on the faculty at Harvard Medical School for more than a decade. Dr. Ji has mentored over 100 scientists, many of whom now lead independent research laboratories worldwide. He serves on the editorial boards of Anesthesiology, Pain, Journal of Pain, Journal of Neuroscience, Journal of Clinical Investigation, and Physiological Reviews. His laboratory investigates GPCR and glial cell mediated signaling mechanisms underlying the development and resolution to pain. So I will now turn things over to Dr. G. Uh, Dr. G, you have to unmute yourself.
Okay, can you hear me?
Yes.
Yeah, sorry for, and let me share my screen then. Can you see my screen? Not yet. Let me stop this. Let me start this
No, right
first.
now.
Let me share again. Yeah. Can you see my screen now?
Yes.
Yes, yeah.
Okay. Thank you very much, Dr. Basu, for your kind introduction. I also want to thank, PRF, for organizing this interesting session on peptides and pain. And as we know, PRF, is becoming a very important source, right, for pain-related research and knowledge. And I really appreciate Becky for her effort. And I will switch gear from MPY to neurotensing, especially now we're focused on neurotensin receptor 1. And we developed an allostatic modulator of neurotensin receptor 1 for optimized analgesic effect in acute pain and chronic pain. So here first I want to introduce our Center for Translational Pain Medicine. And this is a post, and we made, here you can see our faculty, our fellows, our students, So you are welcome to visit us. So our mission is to transform the way we study, diagnose, and treat painful conditions. So now back to introduction to neurotensing. And neurotensing is a certain amino acid neuropeptide. It plays a very important role, widely expressed in the central nervous system. It modulates pain, temperature regulation, and blood pressure oppression. And the anti-nociceptive effect of neurotensing actually has been known for many, many years, pioneer work from Tony Yaksh in 1970 and 80s has demonstrated a very powerful analgesic effect of neurotensin after intrathecal injection. However, in the clinical development of neurotensin is very limited due to its side effects, including hypothermia and hypotension. So neurotensin receptor 1 is known as a high affinity receptor, and which is largely expressed by neurons, whereas the neurotensin receptor 2 is a low affinity receptor mainly expressed by glial cells. And recently, people proposed an interesting idea about allostatic modulator of energy protein coupled receptors. And here you can see and as a GPCR after activation by its ligand, you can see G protein signal, but also and this GPCR activation will lead to a beta arrestin signal, as a negative feedback, which will cause receptor internalization. So the concept of biased allostatic modulator is here. You can see, if it's unbiased, you will get equal activation of G protein signal and a beta arrestin signal. Whereas biased allostatic will cause distinct activation. Here you can see distinct activation of G protein and a beta arrestin signal. So hopefully in this slide you can get more information about allostatic modulator here now, and you can see now that a neurotensin, right, which can bind from extracellular site of GPCR, whereas this allostatic modulator binds from intracellular site. So it's basically on the same receptor, but a very different binding sites. So our collaborators developed an SBI compound. So initially SBI 553 first generation, and SBI 810 is second generation with replacement of methyl, right? And here you can see that in vitro assays for beta arrestin 2, right? And a neurotensin is very potent to induce beta arrestin 2 recruitment. And SBI 810 can also recruit beta arrestin 2. Okay, so this effect can be specifically blocked by neurotensin receptor 1 antagonist. Interestingly, you can see the G protein assay. In this case, is GQ assay. Only neurotensin can induce this GQ activity. However, this SBI compound has no effect and on GQ activity. Actually, SBI 810 can even suppress GQ activity. So here I think you can see and they have very distinct signaling in G protein versus beta arrestin signal. So next, I will show in several slides about an analgesic effect of this SBI compound in acute pain and also in chronic pain. And first, we tested anti-nociceptive effect in naive animals, right? So after IP injection, and you can see the pore withdrawal threshold vehicle no effect, and this SBI compound can produce increase the pore withdrawal threshold and from 1 to 3 hours, and this inhibitor has no effect on the motor function in low-load test or open field test, has no effect on tactile sensation in stick tape test and cotton swab test. So it is specific to inhibit the mechanical nociception. Okay. So next, we test specific mechanisms, whether it's mediated by neurotensin receptor 1 or by beta arrestin 2. So we tested here knockout mice of neurotensin receptor 1 and neurotensin receptor 2, and the effect is abolished in neurotensin receptor 1 knockout mice. And then we also tested the beta arrestin 1 and the beta arrestin 2 knockout mice. The effect is abolished in arrestin 2 knockout mice. So this critical experiment confirms that this SBI compound and induces a mechanical analgesia anti-nociception through neurotensin receptor 1 and beta arrestin 2. Okay. And then we test two acute pain, acute postoperative pain models. One is after pore incision, another is after bone fracture. So in both the models, this compound is highly effective and to reduce mechanical pain and spontaneous pain. And so I will not go through all the details because of time limit. And we also test the chronic pain model after spinal nerve injury, and this compound is also effective. And we compared it with gabapentin, a typical treatment for neuropathic pain. It appears this compound is more effective than gabapentin. So we also tested the spontaneous pain in the CPP test, and you can also see there is inhibition of spontaneous pain, and whereas in the gabapentin can inhibit cognitive function, and this SBI compound does not. So in conclusion, this compound is effective in reducing both acute pain and chronic pain, and without side effects. So next, and we examine the possible cellular a mechanism of this compound. This is a double staining for neurotensin receptor 1 and neurotensin receptor 2 in the spinal cord in the dorsal horn. As you can see, these two receptors show very distinct expression patterns. Basically, neurotensin receptor 1 is expressed by new M positive neurons, and whereas the neurotensin receptor 2 is expressed by non-neuronal cells, especially astrocytes. Okay. And then we also generate a report mice because the antibody may not be very specific. Then we can use anti-HA tag antibody to demonstrate neurotensin receptor 1 expression widely in dorsal horn neurons. Here we want to show some the spinal cord synaptic and mechanism related to central sensitization. So there are two players in central sensitization. One is activation of ERK MAP kinase by phosphorylation. Another is activation of NMDA receptors. And here you can see very low PRK expression under the normal conditions. But if you inject NMDA intrathecally, you can induce very strong ERK phosphorylation. And this phosphorylation is blocked by SBI compound. And through electrophysiology, you can also see NMDA can induce the current, but which is blocked by SBI compound. And this effect is lost in neurotensin receptor 1 and arrestin 2 knockout mice, suggesting that the SBI effect is mediated by neurotensin receptor 1 and beta arrestin 2. And we further examine the peripheral mechanism, and by conducting patch clamp recording in mouse DRG neurons. And as you can see, the action potential filing is suppressed by SBI compound. And on here you can see the expression of neurotensin receptor 1 in DRG neurons, which is very low. The expression is very low, so that's why we use an anti-HA tag. And you can see the expression actually is near the cell surface now. And also and we did the PLA assay to show the interaction between neurotensin receptor 1 and the sodium channel NaV1.7 on the cell surface. And this red wrinkle shows possible interaction of these two molecules at the surface of dorsal horn neurons. Okay. To enhance the translational relevance, we also conducted recordings in human DRG neurons. And as you can see, this compound can also effectively suppress neuron excitability in human DRG neurons. This work was conducted by my former PhD student. Okay. So we also examine the correlation between SBI analgesia and morphine-induced analgesia. As we know, that morphine can produce a reward or CPP, but not SBI compound. And furthermore, morphine-induced CPP can be suppressed by SBI compound, and indicating that this is also possible in inhibiting of opioid receptor signal. So after our paper was published in last May, and John Wood wrote in a very nice preview, and to summarize the action of this SBI compound, basically no side effects, and it can act through both peripheral mechanism and also by modulation of internalization of sodium channel, and also the central mechanism by modulation of NMD receptor signaling and ERK and signaling. Okay, so, and this is my acknowledgement, So here on the left is people in my lab who are involved in this project. And here I think are my collaborators at Duke and at an SBP company and also and at University of Minnesota. So especially I want to thank and the two talented let's say lab members. Dr. Ran Guo is a postdoc in my lab, and Oh Yang, a former PhD student. So and this is my support. Thank you for your attention.
Thank you so much, Dr. G, for that great talk. We'll move to our final speaker for today, Dr. Donald Ian McDonald. So Dr. McDonald is a postdoctoral fellow in the Sensory Cells and Circuits Lab at the National Center for Complementary and Integrative Health, NCCIH of National Institutes of Health. He graduated from the University of Oxford in 2015 with a BA in Cell and Systems Biology, and then earned a PhD in Neuroscience from University College London in 2020, supervised by Professor John Wood. He came to the NIH in 2021 to work with Dr. Alex Chesler and is currently investigating how nociceptors use different neuropeptides to control pain and inflammation. His work is supported by a Branko-Weiss Fellowship and a HEAL Initiative Pathway to Independence Award, the K99R00. So please join me in welcoming Dr. McDonald.
Okay, thank you so much, Paramita, and also Becky for inviting me here. I'm really excited to share some of my postdoc work with Alex Chesler at the NIH. And so I guess without further ado, I'll get started. So I don't need to convince this audience that pain is an important problem. 20% of Americans have chronic pain, but our current therapies are either addictive or ineffective. So I think there's really a pressing need to understand how pain is generated at the cellular molecular level to find new therapies, just like Rurong was showing. So one way the field has really focused on doing this is by focusing on the role of the peripheral nervous system. And in particular, these neurons called nociceptors, which, as we all know, respond to noxious stimuli in the periphery, fire action potentials, and eventually release neurotransmitters into the spinal cord to activate ascending pathways that ultimately drive that pain experience. Now, the field has focused a lot on how these nociceptors respond to noxious stimuli, how they fire action potentials, as a pathway to finding targets that could then silence their activity and treat pain. But another way we might do this, and I think Rurong kind of showed an example of that, is by focusing on how the nociceptors actually use neurotransmitters to communicate with the central nervous system to drive the pain. So in my postdoc, the question I wanted to do is to try and understand what transmitters do the nociceptors use to eventually drive the pain experience. And one of the problems is these nociceptors have dozens of different transmitters. So what I wanted to do is figure out which of these are important, of which of these might be good targets for analgesia. So I think we all know that nociceptors are excitatory neurons. I think there's been a lot of work on glutamate as a nociceptor transmitter. As we know, this is an amino acid transmitter. It acts mostly on inotropic receptors, mainly locally at synapses to cause fast changes in excitability. So, perhaps glutamate is the whole story. Maybe we just block glutamate and we block pain. So I wanted to ask the question, is glutamate the only thing that mediates pain? So to do that, we use the genetic strategy combined with mouse behavior. So obviously, we do lots of different behaviors. Today, for simplicity, I'm just going to show one, which is this so-called ramping hot plate assay, where we measure the temperature at which mice first display pain-like licking behaviors. So in control mice, this is about 43 degrees C, which is basically the heat pain threshold for humans. But what happens if we get rid of nociceptors' ability to release glutamate? So to do that, we knocked out VGLUT2, the vesicular glutamate transporter 2, in TRPV1 Cre lineage nociceptors and asked, how did that affect pain behavior? And we found that knocking out VGLUT2 in nociceptors made the mice slightly less sensitive to heat. And this basically recapitulated findings that were made by 3 independent groups 15 years ago, showing that knocking out VGLUT2 reduces pain. But I think you'll agree that that effect I'm showing you there is not the world's most impressive pain phenotype, right? And in fact, if you compare these mice directly to mice in which we've actually killed off all the TRPV1 nociceptors by expressing diphtheria toxin in TRPV1 nociceptors, I think you can appreciate that clearly VGLUT2 dependent glutamate transmission cannot account for all of pain transmission from nociceptors to the CNS. So I think that begs the question, what else might the nociceptors be using to signal pain in the absence of glutamate? Now, we know nociceptors actually contain many neuropeptides, and these are obviously found in these large dense covesicles, act mostly extrasynaptically. And what's amazing is within each of these dense covesicles, there are actually multiple peptides. Okay, so you can have substance P, CGRP in the same vesicle, and they're released together from the same cells. So to understand what role the neuropeptides were playing in driving the pain, what I wanted to do was come up with a way to actually block their neuropeptide release from the nociceptors and ask how that ultimately impacted the pain transmission. So to do this, we went back to sort of the basic biochemistry and thinking about how these peptides are made, right? Peptides are made in a multi-step biosynthetic pathway. And the last step is catalyzed by a single enzyme called peptidoglycan alpha-amidating monooxygenase, or PAM. And PAM catalyzes C-terminal amidation of most nociceptor peptides. So we hypothesized that if we were to delete PAM, we'd block production of mature neuropeptides, block pain transmission, and ultimately block pain behavior in the mice. So we used our genetic strategy to knock out PAM in the TRPV1 nociceptors. Sorry, my slide has frozen for a second, and asked how did that affect neuropeptide production. So along the top here, I'm showing you control mice and sections of the spinal cord. You can see the nociceptor terminals nicely marked with TRPV1. That colocalizes really beautifully with substance P and CGRP. However, if we look at mice where we've deleted PAM, you can see the nociceptor terminals are still there, marked by TRPV1, but there's no substance P and no CGRP. So this experiment tells us that deleting PAM blocks the production of mature neuropeptides. Great. However, there's one issue with this, right? Which is that the immature, non-amidated peptide might still have some activity at the receptor. So what we needed to do was come up with a bioassay where we could actually measure the ability of endogenous nociceptor-derived peptides to activate their receptors. So to do this, we cultured nociceptor sensory neurons in co-culture with hex cells that I engineered to express the neuropeptide receptor, in this case NKR1 for substance P, human G15 and GCAMP, such that if we come along, we stimulate the nociceptors with capsaicin, the released substance P, it'll bind its receptor and the hex cells will light up, giving us a direct readout of receptor activation by endogenous peptides. So what does that look like in practice? Well, here you can see the nociceptors expressing magenta, and in the control mice, if we stimulate with capsaicin, we see a beautiful release of substance P. Of course, they respond to exogenous substance P, but in the PAM knockout, capsaicin evokes no substance P signaling, and they respond to the exogenous peptide. So what this experiment shows you is that PAM and C-terminal amidation is essential for normal substance P signaling from nociceptors. We also looked at CGRP signaling. We also see this is dramatically reduced in the PAM knockout, telling you that PAM is required for normal CGRP signaling. We also observed a reduction of neuromedin B signaling. However, the neuropeptide PACAP, its signaling was not affected by deleting PAM. So this experiment shows us that by deleting PAM, we could make mice that had a really broad loss of neuropeptide signaling from nociceptors in vitro. So how could we verify this in vivo? So what we did was we took advantage of the fact the nociceptors also release the peptides back into tissues where they interact with the vasculature and immune cells to drive neurogenic inflammation. So we wanted to ask, are the PAM knockout mice, do they lack neurogenic inflammation? So to measure neurogenic inflammation, we used laser spectral contrast imaging to measure blood flow in the pores of mice following stimulation of the pores with the TRPA1 agonist mustard oil. You can see in the control mice, there's a really nice increase in blood flow, and this is strongly abrogated in the mice where we've knocked out PAM in the nociceptors. So this experiment tells you that in vivo, we've had a reduction in neuropeptide signaling too. So then the million-dollar question is, how did this affect pain transmission? So we took our PAM knockout mice and asked, how did this affect their response to noxious stimuli? Well, this is what we found. The mice had a slight decrease in their sensitivity, but again, this is not a really dramatic phenotype. So at this point, I start to get worried, right? I need to finish a postdoc and get a job, and here we are with these piddling phenotypes. So I wondered if perhaps what's going on is that, we've made a nociceptor-specific knockout, but it's still constitutive, right? The mice are lacking it from birth, so we thought, well, maybe there's some kind of compensation. So we did bulk RNA sequencing comparing the PAM knockouts to controls to see if there was any transcriptional dysregulation. And we found that there were two genes whose expression was changed. In the TRPV1 PAM knockouts, they had more Cre and less PAM. So clearly, there's no transcriptional compensation going on here. So how can we explain this? Well, arguably this result wasn't that surprising because I'd already published a paper showing that mice lacking substance P and CGRP had pretty normal pain. And indeed, if you look at the TAC1, CALCA, CALC-B triple knockouts that we recently generated, we can see that these mice also essentially phenocopy the PAM knockout and have a really, I'd say, marginal loss of pain. So I said before that PACAP signaling was unaffected by PAM deletion, so we also checked whether the PACAP might be the key transmitter, but in fact, the PACAP knockout mice had completely normal pain. Okay, so at this point, you're getting really worried, right? You've made PAM knockout mice that lack most of the peptides, their pain is normal. You've made VGLUT2 knockout mice that lack glutamate transmission, their pain is pretty normal. So, what on earth is actually carrying that pain signal from nociceptor to CNS? Well, we wondered if perhaps what our data is telling us is that these transmitter systems are both on their own sufficient and that they might be mutually redundant. So to test that, what we did is we made a PAM VGLUT2 double knockout mice that lacks both peptides and glutamate and asked how did that affect pain. And what we found was something quite remarkable. I'm just waiting for it to appear. You can see there that the PAM VGLUT2 double knockout has a dramatic loss of pain sensitivity with many mice going to cutoff. So this tells you that you get a synergistic analgesic effect when you knock out both the peptides and the glutamate. And this isn't specific just to this assay. If we look at the conventional static hot plate, again, you only get dramatic analgesia when you get rid of both glutamate and peptides. Same things from the Hargreaves assay. Same thing again for the dry ice test, only the double knockout has a phenotype. And same thing again for the capsaicin assay, only the double knockout has a phenotype. So what these data are telling us is that you need to get rid of both glutamate and peptides to block the pain, and that both these transmitter systems are degenerate or redundant. And it could be, you might be thinking, perhaps one of the problems is you're using this weird PAM strategy that no one's used before. So we also wanted to test using a more conventional sort of neuropeptide-specific knockout strategy. So here I'm showing the ramping hot plate response, VGLUT2 knockouts, 45 degrees, TAC1 knockouts about 44 degrees. If we make a mouse that lacks both TAC1 and VGLUT2, i.e. lacks glutamate and substance P, you find these also have a synergistic loss of pain. So altogether, I think these studies really show us that nociceptors are using multiple neurotransmitters to drive pain, and they rely on these multiple mutually redundant systems using both glutamate and neuropeptides to ultimately drive the experience of pain in the brain. So thank you so much for your time. I just want to thank my PI, Alex, Rakshita Balaji, who was a postback who helped a lot with this study, the Chesler Lab, and also my K99 mentors, Nick Reba and Isaac Chu. I'd also like to thank my funders, and in particular, the HEAL Initiative, who supported my K99. I am on the job market, so if anybody knows of any great jobs going in the pain field, please let me know. Thank you.
Awesome. Thank you so much, Dr. McDonald. And I would like to give a big round of applause to all our speakers for your great talks. So now we're going to open, we have just like 3 minutes. So we're going to open the floor to questions. So audience, please feel free to, wait a minute. I think Stephanie, Let's see. Okay. Okay, do I see? I don't see any raised hands on my end. Okay, let's see. Okay. Yeah, it's just me. Okay, so if you have any questions, you can type it in the chat box. Okay, so we have a question for Dr. McDonald. This is from Melissa. So her question is, did you look at the mechanical sensitivity at all?
Yeah, so I mean, the simple answer to this question is that I mean we did, but we're using the TRPV1 Cree to target the great majority of nociceptors, and one of the problems is the TRPV1 Cree DTA mice, for example. These mice don't have any deficits, really don't have much or if any deficits in mechanical sensitivity. So because that Cree line spares, I guess, enough mechanonociceptors and enough LTMRs to sustain mechanical responses. So essentially, all of our mice have normal noxious mechanical sensation by virtue of the Cree line we used. So yeah, that's why we focus on chemical and thermal pain because they're relevant to the cell population we're targeting.
Thank you. So we also have an applaud just to let you know our speakers from Stephanie Sutherland. We have another question for you, Dr. McDonald. It's from Christopher Conrad. It said substance P has shown inconsistent results in clinical trials for pain relief in humans, despite its significant role in pain signaling in animal models. Can you comment on the difference in translation?
Yeah, thank you for the question. So I would argue that the literature from animal models on the role of substance P in pain is also rather inconsistent. And I think that the talk I just gave probably gives one explanation for that, which is that substance P signaling can be compensated for by other transmitter systems like glutamate. I expect that explains part of the reason. I think another important point is that substance P clearly plays an important role in neurogenic inflammation, and that appears to contribute to some forms of pain in mouse models, such as the post-surgical pain in mouse models. So it's possible we just need to be very careful about the types of pain conditions we study in the mouse and then in the human to make sure that they are the pain conditions that may or may not involve substance P and potentially neurogenic inflammation.
Okay, so we have a question from Brian Room, and I'm going to let him speak. Go ahead, Brian. Can you hear me, Brian?
I think he's muted.
Yeah, I just asked him to unmute. You have to unmute yourself, Brian, to ask the question. Okay, while you're figuring it out, we have another question for Dr. McDonald in the Q&A. Does each neuron use multiple strategies, that is multiple neuropeptides, or is that each neuron uses different neuropeptides from the others?
Yeah, I mean, I think, so, you can explain these data by saying the nociceptors are homogeneous and they all release both glutamate and the peptides or rely on both together, or you can explain it by saying there are some nociceptors that use only neuropeptides, and there are some that use only glutamate, but both contribute to the behavior. I don't think these experiments don't distinguish between those possibilities, but of course, the vesicular glutamate transporter 2 is basically expressed in all nociceptors. CGRP is expressed in essentially all nociceptors in human and mice. Substance P is expressed in the great majority. So I think the former explanation is probably the most likely, but yeah, these experiments don't distinguish those two possibilities.
Got it. So continuing with that, Alni, his question is that do you have any thoughts on how the relative roles of glutaminergic and peptidergic signaling from nociceptors may change in injured or chronic pain conditions?
Yeah,
For
so yeah, thanks for the question. Yeah, so I would say that this study that I presented was really, was concerned with the question of, you know, at baseline to drive the sort of survival related acute pain signals, what transmitters do they use? I think it's quite possible that, for example, that different subsets of nociceptors that might be more reliant on CGRP, for example, come into play to drive certain types of chronic pain. I mean, clearly, CGRP is an essential transmitter for many people that experience pain related to migraine. Although I think it remains unclear exactly what the target of CGRP is. So I think that's a question I would say I'm really excited to investigate in the future, is to try and understand the role of the different transmitter systems in specific diseases. Because I think that's really the pathway to get away from, you could have redundancy in the sort of survival related pain, but you can imagine if we want to create specific types of to create specific analgesics, we want to look for the molecules involved in specific pain states.
Alrighty. So we have two more questions, and after that, we are a little bit running over time. It's all for Dr. McDonald. So first is, did you look at the CGRP, substance P, glutamate knockout influenced inflammatory responses in the periphery beyond blood flow, that's edema or heat? This is from Lauren Donovan.
Yeah, so that's something we're actively investigating at the moment. I'm very interested in how the different neurotransmitter systems interact with immune cells and also vascular cells. We have developed assays to combine like optogenetics with measurements of blood flow or heat, etc., to start looking at this in, I would say, a much more controlled fashion, to identify how specific types of nociceptors control those responses and what molecules they use. So we're very interested to look into that.
Thank you. Did you notice any sex differences or age-related differences in the role of neuropeptides in pain transmission?
So all the experiments were performed on male and female mice, and we saw no overt or obvious differences between male and female mice. All the experiments were performed on adult mice, within a certain age range, right? They're at least 8 weeks, and we don't go much older than 16 weeks. So that's something, could be very interesting to look at in the future, but not something we looked at in this study.
Perfect. So, well, since we are running out of time, it's already 5 minutes over. If you have any other questions, so please reach out to Dr. Taylor, Dr. G, and Dr. McDonald. So I really appreciate all of your participants. And once again, thank you so much to all our three speakers for the great talk. And thanks for joining the PRF First webinar. Thank you so much.
Yeah, thank you. Yeah, this is a great session, a great start of the year.
Thanks so much.
Take
you.
care.
Thank you.